welcome to today's lecture. I hope all of you watching are keeping healthy and are keeping in as good spirits as possible as we navigate this unprecedented situation. And so I want to begin by just saying that I'm a data scientist and a professor working on some research in some specialized areas, specifically in protein structure prediction, and which is, of course, itself also a sub-area within computational biology. And so with the ongoing situation, the coronavirus pandemic has rightfully received a lot of attention from the scientific community as we all try to find answers and solutions. And I would also say there's a lot of ways for statisticians and data scientists in general to chip in to these efforts. Though today I will focus on one relevant aspect that I am most familiar with speaking on from a data science perspective, and that would be in regards to protein structure. And so don't worry, I will not assume that you know anything about proteins. So in today's lecture, I will give you some background on how proteins play a role in this, the way this virus works. And I will mention some of the problems that the medical, scientific, and computational communities are facing right now and trying to tackle as a result. And along the way, I also want to tie in some of my own thoughts from my own research experiences working in protein structure as well. Okay, so let's get started. For live viewers, if you have any questions, please type them in chat and I will try to answer them as we go along. Okay, so here's a few pictures of some proteins. We can think of proteins as machines of life, as they play such crucial roles in so many biological processes. And so here I've shown you 4 examples of proteins. So on the left there's rhodopsin, and so that enables you to see well in low light conditions. So it's a protein that works in our eyes. Next we have hemoglobin, which we know is a very important protein for blood transport. Next there's myosin, and that's a protein that's crucial for our muscles to contract. And finally on the right we have pepsin, which is an enzyme used for digestion. And so I trust that all of these proteins are working correctly in your body as you watch me speak. Okay, so even if we're not consciously thinking about it, these proteins are doing their jobs in our bodies and keeping us going. So the other thing that I want to point out is they all have very distinct shapes, as you can see in these pictures. Okay, so these shapes, or what I will call their 3-dimensional or 3D structures, are very much associated with how they're able to carry out their functions. Okay, so their shape particularly allows them to bind to the relevant molecules so that they can do their job. Now, on the bottom right, I mentioned that we're able to know these structures because they have been deposited in a public database on the internet, and that's called the Protein Data Bank, or the PDB for short. Okay, and so in just a little while I will explain how those structures actually get determined. But before I go there, I want to show you another example of a protein structure. So here's another picture of a protein. Now, in contrast, this is an example of a protein that I certainly pray is not in your system. Okay, this is, in fact, the spike glycoprotein, or the S protein, that is a crucial part of the coronavirus, and it is what allows the coronavirus to gain entry into host cells. Okay, and so this is a particular structure that has just been determined about a month ago, as scientists started working around the clock to gain as much information about this novel 2019 novel coronavirus as quickly as possible. Now, there's a few distinct regions here that are color-coded, and I want to point out one really important region, and that is this part that is highlighted in green. Okay, so I've just drawn an ellipse around there. Okay, so this is this portion that you can see is sort of sticking out from the rest of the protein. Okay, so this is known as a spike protein, in particular because of the functionality of this green part. Okay, so it has a name, and it is called the receptor binding domain. And actually, it has two possible structural conformations. So here, the one that's shown is it's the what's so-called its up conformation, in the sense that it has in a sense it is folded up from the rest of the protein, and this is the form of the structure that facilitates host entry. Okay, so the reason it's called the receptor binding domain is because this is the part that latches on to host cells and allows the virus to gain entry. So it's ready to attack in this conformation. Okay, so it also has a down state, and so this receptor binding domain sort of works like a hinge. So depending on what it is currently at, it'll be in either the up or the down state, and the up state is slightly less stable, but it's in that state as it's ready to attack a host. Okay, so this protein structure is called also known as the pre-fusion conformation of this coronavirus spike protein, and we call it pre-fusion because this is before it latches onto the host receptor. In particular, the specific cellular receptor that this RBD binds to is the angiotensin converting enzyme 2. Okay, so also known as the ACE2. And so, of course, the structure of this protein also evolves once it attaches. And so, in a follow-up study from this one, several weeks later, scientists also determined how the complex of these two things together, so the spike protein along with the ACE2, what that looks like as a joint complex. So that structure has also now been determined. Okay, so I do want to point out here that in fact, the ACE2 is exactly the same cellular receptor that the original SARS virus also binds to. Okay, and so of course, one of the first things that that medical scientists investigated, well, we have antiviral interventions for SARS, and so of course, one of the first things that was tried was to see whether any of those antiviral interventions that targeted SARS, whether they worked for the novel coronavirus. And it turns out that they do not. They do not bind to the novel coronavirus. And Well, why is that? Well, it turns out as much as they're related, the SARS viral protein and the novel coronavirus protein have enough structural differences in this spike protein such that the same antiviral treatments do not work. Okay, and so therefore there's worldwide efforts ongoing right now that involve designing new therapeutics that can bind to and disable the RBD of the novel coronavirus. And so that's the key question that I just put on the screen here is, well, how can we neutralize this protein? And so one way is to block the RBD by, for example, designing drugs or proteins that the RBD would bind to instead of attacking the host cell. Another reason that we've now learned why the novel coronavirus is more deadly in some sense than SARS is that this receptor actually binds more strongly to the host cell in the novel coronavirus compared to SARS. So that's another structural difference that scientists have now elucidated. Okay, so all of this was published very recently. In this 3D structure was published in Science just about a month ago, and so that can be found in the reference that I've cited down here. That's where the picture comes from, the March 2020 issue of the Science journal. Okay, so next I want to give a little bit of background so you understand how these different pieces of, you've probably heard about genome sequencing, and then we talk about protein sequences, and then protein structures, and finally their function. So as kind of a point to keep in mind is this idea of studying protein structure and their relationship to sequence is now a 5-decade-old problem, and so the key thing I want to mention here is that so one very important discovery made in the 70s is that the sequence of amino acids that compose a protein is generally sufficient for determining what is its stable three-dimensional structure. Okay, so in other words, there's, generally speaking, a correspondence between sequence and structure. Okay, and the reason that's important is because the structure, as mentioned in some of my earlier examples, is very much tied to the ability of the protein to function. Okay, and so in fact, and that's why anytime our body produces, say, hemoglobin protein, it's composed of the same sequence of amino acids, and it folds into the same 3D structure, which is what gives it consistency in being able to carry out its job in our bodies. Okay, and so this was a very important discovery, and so the scientist that this was attributed to, Christian Atkinson, was awarded a Nobel Prize for this discovery in the 70s. Okay, so on the left-hand side, you see the whole process of looking at protein structure starts off by first determining, well, what is the genome sequence that codes for the protein. And so that's where work in terms of genome sequencing, and now we have high throughput sequencing, comes into play. The first step is determining what is the amino acid sequence. Of course, we have much more sequence data than structure data, as I will explain in a moment. But that's the first step in being able to study protein structure and function. Okay, so when I say protein folding, I'm just again to give a very brief background. I'm talking about the process by which a protein, so we can encode it or we can represent it as a sequence of amino acids. So there are 20 different unique amino acids, so they can be represented in terms of letters representing each distinct amino acid. So say here is an amino acid sequence corresponding to a protein. And so folding is the process by which it forms a stable three-dimensional structure. Okay, so the structure is the one shown on the right here. Okay, so the key discovery by Anfinsen is that in most cases there is a direct correspondence. So anytime you see this sequence on the left, it folds into the structure on the right, and it's a stable structure. Now you see in this ribbon diagram, there's a few key features. So just to point them out briefly, we see there's kind of three color-coded parts, and they correspond to what we call secondary structures within the protein. So the helices are called the alpha helix, and then you see these strand-like features. They're called beta sheets, and then these connecting regions, which tend to have quite a flexible structure, are called coils. So let's take the novel coronavirus as a specific example. Okay, so first we need to know its sequence. And so thanks to current technology, all we need now to be able to determine the genome sequence is simply to get a swab of, say, from the nose of an individual who is infected with the coronavirus. And so that's enough of a sample such that we can extract RNA from the sample, and therefore that allows us to determine the underlying sequence of letters that code for the key viral proteins. Okay, and so after scientists have done that, then they can take what is now known to be the amino acid sequence for the key proteins of the novel coronavirus, and they can build these proteins synthetically in a lab environment, and from there they can study the structure. Okay, so just a quick overview of some lab techniques that are available to determine structure. So traditionally, the way to do that is via techniques called either X-ray crystallography or nuclear magnetic resonance, so that's known as NMR. Okay, and this was first done by Kendrew and his co-authors, and this kind of technique was first used and published in a Nature paper in the late 1950s. And so that technique has, of course, been refined over the years, but nonetheless, it is considered a relatively expensive and time-consuming procedure, and you certainly cannot do it for all proteins. In fact, it does not work for all proteins. And as mentioned, there's far more sequences that code for proteins than we can possibly analyze in a laboratory setting using these techniques. So to give a specific example, so for the S or spike protein, the spike glycoprotein for the novel coronavirus that I showed you, so that was actually solved in a lab environment in what we would consider record speed. Okay, so it took only thanks to efforts around the world. The first lab to be able to do it was a team of scientists at UT Austin. Okay, so they were able to do this in just 12 days, which is extremely fast for structure determination. And they used a technique called CyRO-EM or cyro-electron microscopy. And so they were able to get this reviewed and published. So this 3D structure that I showed you earlier was published in Science, as I showed you, on February 19th. Okay, so this was considered quite a remarkable success in lab techniques of being able to find the structure very quickly. So on the flip side, so there's lab techniques, but there's also computational techniques. And in fact, ever since computers were developed, people have asked us this question: well, since there is essentially a mapping from sequence to structure, can we use computer algorithms to correctly predict the 3D structure from the sequence? And so that's been a fundamental problem in computational biology for the last few decades. And there's certainly been quite a lot of progress. And so, in fact, this is recognized to be such an important problem that if it can be solved computationally, it would have huge ramifications for modern applications like protein drug design. So two of the early pioneers, Martin Karplus and Michael Levitt, two early scientists that pioneered computational methods for protein folding, were in fact awarded a Nobel Prize in Chemistry in 2013 for their contributions to these efforts. So I mentioned, there's significant applications in protein drug design, and so specifically, if I want to link it to the coronavirus, so this would be one example of a question that would be relevant. So can we design protein sequences that can latch onto the receptor binding domain of the spike protein? And specifically, we want to do that in prefusion form, in other words, before it attaches to host cells, and thus, therefore, neutralize it. Okay, and so to do so, why is computation helpful? Well, we want to be able to efficiently narrow down, well, what are potentially useful protein sequences that could accomplish this task? And so essentially we're asking, well, what are some candidate sequences that could fold into a 3D shape that could potentially have a neutralizing effect? So in simple terms, that's one way I can describe it. Okay, and so that'd be very useful. Now, since there are so many possible sequences that we could build, computational algorithms could help narrow down what people actually need to try putting together in the lab. Okay, so in that sense, computers and algorithms can play a really important role in this drug development and drug discovery process. Okay, so one underlying principle that is kind of a useful way to conceptualize protein folding from a computational perspective is what we call the energy landscape, and I'll come back to that in the later part of the lecture. So here's a stylized diagram that shows the energy landscape for protein conformations. Okay, so this is of course kind of an idealized picture, but it does show some of the key points I want to mention. So we can think of that the protein itself, when it's actually folded in its stable structure, stabilizes near a low energy state, and so you see that at the bottom of this energy well. Okay, and so I'm going to use the notation cap H to represent what we call the energy function. So you can think of the stable conformation at the bottom here is generally associated with the lowest energy state that the sequence of amino acids could assume in 3D structural form. Okay, so corresponding to an energy function, we can write down a corresponding probability distribution. Okay, so I've denoted this pi x. So this is what we call the Boltzmann distribution corresponding to the energy. So we can take the energy function, and exponentiate it. So raise it e to the minus energy. Okay, and then you can scale the energy by any constant. For example, the Boltzmann constant Kb and a temperature. Okay, and then Zt below is simply the normalizing constant. So that is what allows this to be a proper density function. So it's so we get that by summing over all the states according to their probability, such that it's a normalizing constant for this probability distribution. Of course, in practice, this conformational space is extremely large, and so there's no hope of computing this normalizing constant Zt, or even enumerating the space of possible conformations. But conceptually, we can think of that when a conformation is at a high energy. So, for example, these conformations up near the top here, which have much higher energy, would be considered part of, say, unfolded states or incorrectly folded states. And so, essentially, if we conceptualize this in looking at this from a computational and data science viewpoint, we're trying to find of all the possible conformations, can we locate the one that would have the lowest or minimum energy using a computational approach. And so to do that, I mean, it's conceptually simple. It's like a energy optimization problem, but it's quite difficult because there are two major bottlenecks or challenges that make this very hard. And first, we need a way to search or sample the space of conformations, and that's hard because of the high dimensionality, and it's also a very rough and multimodal energy surface. And secondly, in order to do this well, we also need an accurate energy function. And so most energy functions that are used for computation are approximations at best. And so to be usable in an optimization procedure, they also need to be as accurate as possible while still being computationally feasible to use. So that's how we can think of, that's one way we can think of computational protein folding as a very high dimensional optimization problem, where statistical strategies such as sampling play an important role. Okay, so in practice, so suppose now, so let's go back to now we have a new protein sequence that we have just obtained. And so, for example, when researchers first got to know what the sequence of the coronavirus spike protein was, well, they wanted to first build before labs were able to produce the real 3D structure. People were already on it from a computational viewpoint. They wanted to build a structure prediction for this new unknown protein that we needed to study. So the way to do that in practice is to make use of known structures in the protein data bank, so that have already been deposited to help build an initial three-dimensional structure prediction. And so key to being able to do this is again a general principle that similarities in sequence often correspond to structural similarities as well. So to take as an example, here is, suppose we have a sequence of amino acids that we want to build a structure prediction for. Okay, so how it is done is, well, first you would take, because of this general principle, we would first take this sequence of amino acids and try to align it with sequences in existing databases. Okay, so essentially we're looking for existing protein sequences that have significant similarity in sequence to proteins where we already know their structure. Okay, and after doing that, so here's an example of a picture. So on the top is the new sequence, say, that we're trying to study, and then on the subsequent rows are some matches that are output by a sequence alignment tool. So that tells us what are some sequences that have some sequence identity and similarity with this new protein that we're trying to study. And so, of course, there may be parts of the sequence that don't align very well with those of existing proteins. So here we see there is a segment here that, for example, does not have very much alignment with existing proteins. So that's a weak alignment region. And so essentially, to build an initial structure, in simple terms, researchers can stitch together the 3D structural information from the matches and build a 3D prediction. Now, not surprisingly, parts of the structure where there are not very much alignment information to help inform tend to be the least accurate. Okay, so in that particular example, so here are the two structures. On the left is the true lab-determined structure that was later determined by crystallography, and on the right was the 3D structure that researchers were able to build based on sequence alignments and stitching together a structure. So you see, there's, broadly speaking, you see these kinds of methods do a pretty good job. You can see, broadly speaking, there's a remarkable structural similarity between the predicted one and the lab-determined one, which was released later. And so I just highlighted here in green. There's one part that seems to be sticking out and doesn't quite look like the true structure. And not surprisingly, that's also the region of the protein where there was not very much sequence alignment. And so that, in fact, that's the part where we identified in that diagram on the last slide, where there was a weak amount of alignment. Now, so that's in general. Now, I want to talk specifically about how this was applied for understanding the spike protein of the novel coronavirus. Okay, so here I've just shown an excerpt of the novel coronavirus protein, and I have specifically just excerpted the green part, and so this is the RBD, the key part, the receptor binding domain, which attacks host cells. So on the top row is the sequence of this domain in the novel coronavirus, and then just in a similar way, we've put the sequences of two aligned proteins. So there's two here. One is the SARS protein, and then below that is another coronavirus protein. It's a bat coronavirus. Okay, it's called the R-ATG13. Okay, the reason it's so of course we know the SARS virus did attack did sicken humans, but the bottom one was a bat virus, and so we do know that it's a related family of proteins. So in fact, if we look carefully at these sequences, in fact, the bat coronavirus protein has the strongest sequence similarity with the novel coronavirus. In fact, it has 98% of the amino acid sequence is exactly the same. Okay, but nonetheless, the SARS coronavirus protein is also relevant because that's one that has been heavily studied before by scientists. Okay, so here is the three sequences aligned against each other, and so you can see in the asterisks here that there are definitely quite a lot of positions where all three sequences agree, and so the positions where you don't see an asterisk are where the sequences diverge in the sense that they have different amino acids in those positions. So you see there are some regions of divergence among the three amino acid sequences, especially on the bottom row. Okay, so of course there are other homologous sequences, in other words, other sequences that can be aligned with the novel coronavirus. And so these were used by computational biologists to build a preliminary predicted structure before the 3D one was made available. Okay, and so let's just show you how well that worked. Okay, so here just focus on the binding region only, the RBD. So this was done. This was a structure prediction released by researchers at the University of Washington. Okay, so this was done using their modeling set of tools called Rosetta. So using those tools, they built a preliminary 3D structure prediction for the RBD. So that's the part that's highlighted here in blue. Okay, so that's the RBD section in blue. And so later, when the true structure was published in the science paper, then that's the part colored in tan. And so in this picture, they are overlaid on top of each other, and you can see, well, there is quite a bit of similarity between the predicted structure and what was later determined to the true one. Okay, so we can see the value of these sorts of computational algorithms in giving us a head start in knowing what are we dealing with in terms from a structural level of this protein. So that's quite a success in terms of applications of these sequence alignment to structure prediction tools. Now, an earlier point I mentioned was that there is a general correspondence between similarity in structure versus similarity in sequence. And so we saw that there was some similarity in sequence between the SARS RBD and the novel coronavirus RBD, but that there were also significant regions where there were differences. And so here, from the same science paper, the researchers superimposed the RBDs of the two proteins. Okay, so here they're overlaid on top of each other. The SARS RBD and the in gray, and the novel coronavirus RBD in green. Okay, and so we see that there is, in fact, quite some noticeable structural differences between these two RBDs, and that's not surprising because, as we saw when the sequences were aligned together, there were definitely parts where there was not a strong amount of sequence similarity, and so we see those differences also reflected in this picture that shows differences in structure. In particular, one region to point out here is there is on the right hand side here is a loop region that seems to differ substantially between the two spike proteins. Okay, and so that gives us some intuition behind why there's enough structural differences that existing ways of targeting the SARS RBD does not work for the novel coronavirus RBD. Okay, so that's one aspect that we now know about, which is the 3D structure of a particular sequence corresponding to the novel coronavirus spike protein. Okay, so we now have a 3D structure which scientists can now work with for developing drug interventions. But there's kind of one twist to this narrative and this story, and that's the fact that the virus itself is not static. Okay, so it's what we mean by that is that the viral genome continues to mutate as it is transmitted. And so, in other words, there are mistakes and substitutions and perhaps deletions or additions in the viral genome as it's passed from one person to another. Now, most of such mutations will not have a significant impact on the amino acid sequence of, say, the key proteins that allow the virus to attack hosts. But it is worth pointing out that currently there's already seen in the database that there's already at least 10 variants in the amino acid sequence seen for the spike protein itself. Okay, and so these variants can be seen and obtained from a global database. So the website's here, and so we have the 3D structure for a particular sequence, and so it can be also of interest to investigate, and it'd be useful to examine potential impacts of how mutations, so changes in to local regions of amino acid sequence, and how they could actually play a role in affecting how the 3D structure and function of say the spike protein might also vary as this virus mutates over time. Okay, and so here is a good illustration of this sort of idea. And so here we've taken this screenshot from this website called NextStrain, and so this website tracks the change in the phylogeny of the viral genome as over time and also over space. Okay, so so the key takeaway from this picture is noting that in fact, yes, the viral genome has been changing even as the virus has been spreading to multiple countries. And so what we notice, so this is kind of color coded by region. And so you notice that, for example, in North America, the genome variant of the coronavirus that we're seeing is spatially correlated. Okay, so in other words, the variant of the coronavirus that's spreading around here in North America is more similar amongst themselves, and in fact, there have been some substitutions in the sequence compared to the version, for example, that was spreading in Asia. Okay, so you can see there's a lot of spatial correlation, and there's clustering by region. So anyway, this is a good illustration of how this viral genome is changing over space and time. Okay, so here's a quick recap so far of this first half of this lecture. So kind of a couple of key takeaways here is that there's hopefully you see by now that there's ample opportunity for data scientists to be involved in tackling even in this very specific sub area, which is the structural biology aspects of the problem. And so I've mentioned two examples of such directions. One was developing methods to identify promising amino acid sequences in terms of how well they would work for treatments, for example, to block the receptor binding domain. And the other one, which is related to this idea of mutations, is to quantify uncertainty associated with possible changes or potential changes in the viral genome, and how they could be associated with changes in the structure and function of key proteins of the novel coronavirus. So these are, of course, just two examples of research directions. And there, of course, can be many more. So I just want to spend the rest of today talking a little bit about this second point, so quantifying uncertainty. So in other words, when there are relatively small local changes to an amino acid sequence, can we assess their potential impacts on the 3D structure of, say, the key proteins. And so in the remaining time, I'm going to outline how we can take some statistical methodology, specifically sequential Monte Carlo methods, to help study and tackle the second problem. Okay, so I will outline some methodology that's been adapted from some of my own research. So this was published a couple years ago in one of my earlier papers. Okay, so this part will start to get a little bit technical and in terms of the statistics, but I just want you to be able to catch some of the big ideas and how we can adapt some of these ideas to help study this second problem. And so these are some ongoing research thoughts that I am currently investigating myself. So let's get into that. Okay, so here is the scenario I want to focus on. So suppose there are several changes to a local region of an amino acid sequence. Of course, as mentioned, we do not have laboratory determined structures for assessing the impact of all of these mutations. We currently have one, or there may eventually be more, but one main 3D structure to work with for the spike protein. And so we want to see if we can take computational methods to assess the potential impact of these mutations. So to boil it down, we want to build structural predictions for, say, the local region or segment, as what I will call, of the protein that is most likely to be affected by the changes in amino acid sequence. And really what we want to do is, we want to quantify uncertainty in what is the space of possible conformations that the mutated portion of the protein could take after those changes. But it's still not particularly easy as a problem even on a local scale, because the three-dimensional space is still highly multimodal, and the dimensionality is quite large. Okay, and so methods from Monte Carlo sampling, and specifically I'll briefly introduce a sequential Monte Carlo method, can be useful for tackling this, because we can adapt such methods to stochastically minimize the segment of an energy and efficiently explore the plausible set of low energy three-dimensional conformations that the mutated region could potentially have. Okay, and so remember in the picture I showed you several slides ago, we were talking about the energy landscape. And so remember, the protein tends to stabilize in a low energy conformation. And so this is a methodology which we can apply to help find plausible low energy conformations. Okay, so that's how it's related to the energy landscape that I showed you earlier. Okay, so here's how we could frame this problem from a statistical point of view. So suppose we are interested in a continuous segment. Let's say we have L amino acids. We want to optimize and find low energy possible conformations. And we're going to do that guided by an energy function, which we'll call H, that governs the goodness, and which we'll use to assess the goodness of possible conformations. Okay, and so we're going to let X denote what we call the free dihedral angles of the segment. Now, I think it would be useful here, so let me spend maybe 2 or 3 minutes. I'll go over to the board and give you a very, very short crash course on protein geometry, so to help you understand this a little better. So here on this slide, I've defined several angles. Okay, I call them phi, psi, omega, and chi. And so for each of the amino acids in this sequence, we would want to determine what are possible values for these particular angles. Okay, so I'm going to go over the board here and just quickly diagram what that looks like. Okay, so here is a quick crash course on protein geometry. So when I was talking about these amino acids earlier, we can think of the protein as a backbone, and that's what's plotted in those ribbon diagrams. So what are the atoms that are at the atomic level? What's actually happening? Okay, so here is what we call the atoms making up the backbone, which gives the protein its structure. So it is a repeating sequence of atoms. So one amino acid would have these are the four key backbone atoms. There's a nitrogen, carbon alpha, carbon, and oxygen. Now, so linking together two adjacent amino acids, there's a peptide bond, okay, and so here we have another amino acid. So we have another N, C alpha, C, and O. Okay, and so this over here is what we call the peptide bond. Okay, and so that links together each of these letters in the amino acid sequence. Okay, so this sequence continues on both sides. Okay, and so one way to think of this geometrically and in three-dimensional space is to look at specific angles that govern the positioning of these atoms. Okay, so there are three key angles that I want to point out, and so they're what we call dihedral angles. So they're essentially the angle of rotation on these bonds. Okay, so over here, so essentially you're looking down the axis of nitrogen and the carbon alpha, and so that's the angle that we call phi, and then there's another angle for the next bond, and that's what we call psi, and then this bond also has an angle, which is the one we call omega. Now, so those are what we call the backbone dihedral angles associated with the protein. So therefore, each amino acid has these three angles, and so we want to look at possible conformations or possible positions of these backbone atoms. We would look at we would want to sample values for these three backbone angles. Now, what makes each of the 20 amino acids distinct from each other is what we call the side chain. So these are functional groups that stick out from the carbon alpha. So each of the 20 amino acids has a distinct side chain. So let's say there's a side chain here and there's a side chain here. Now, of course, for the atoms in the side chain, there's going to be another set of angles that we can use to parameterize the positions of the side chain atoms, and so those are the ones that we call chi. All right, so therefore, to view this as a statistical sampling problem, essentially we are looking at the distribution. We're looking at the distribution of these sets of angles. Okay, so one set of phi, psi, omega, and chi for each amino acid in the region of interest that we want to explore conformations for. Okay, and so we want to sample values of those angles according to a probability distribution. And so what probability distribution? Well, it's the one that's determined by the energy function H. Okay, so that's a very quick crash course on protein geometry. So now let's go back over here and continue. With the slide. Okay, so now I've explained what we mean by the free dihedral angles of the segment. So we have this quadruple of angles for each amino acid in the segment. And so back to the main task. Well, we want to find the minimum energy and optimize this energy function, and which we can view using the equivalent Boltzmann distribution, which I showed you earlier. Now, how can we think of energy functions conceptually? Well, there's two key parts to it. So the energy function essentially helps us determine whether a proposed conformation of a protein is a realistic one or not. And so here are the two key components. So this is quite simplified, but good enough for our purposes. So there's one component that governs the energy of the dihedral angles. Okay, so those are the angles I just illustrated to you. And so the second component is in regards to the energy of atomic interactions. Okay, so as I showed you, the protein structure is really composed of a large number, potentially thousands of atoms that form that stable conformation because they have a favorable atomic interactions in that conformation. And so that's modeled in the energy function with a component that governs the pairwise distances. So in 3D space, we look at the distance between each pair of atoms, okay, and then we calculate energy scores, and generally we put them together to give an overall score for the energy based on the distance interactions. Okay, so here's an example of how these two energy components might look like. So on the left-hand side is, say, an empirical density of some of these backbone angles, so phi and psi. So this is simply an empirical density, and so plotted such that the darker areas have a higher density. And where did we obtain this information? Well, we simply went to the protein data bank and we extracted the angles seen in real protein structures, and basically this is just a 2D empirical histogram of the density. So this can be used to form an energy for the angles because generally, if we were looking at a new unknown structure, the regions of the angle space which are more likely would be, well, the regions where we saw more of those angles in existing structures. Okay, now what about the distance part? How do we know whether atoms are at an energetically favorable spatial distance apart from each other? Okay, so for example, the energy function would help us calculate the effect of atomic clashes. Okay, so here is, suppose we are focusing on exploring conformations of the red segments. Okay, so stylized depiction in those red balls. And this would be an example of a bad conformation because down here, these red atoms are too close to the yellow one, and so this is not a realistic conformation. And so this would be eliminated by a good energy function would not allow such a conformation. Now, because we're focusing on local regions, it's also important that the energy function helps us assure that we have a properly connected protein backbone. Okay, so here is another example that this would not, again, be an unrealistic conformation because the lengths between the bonds of the atoms is pretty much fixed, and so this is an unrealistic conformation because this red atom doesn't connect properly with the rest of the protein at the end. So the role of the energy function would be to eliminate conformations like these. So this gives you a sense of how the energy function works to separate plausible atomic spatial conformations from ones that are not good. Okay, of course, the energy function does a lot more than that, but simply put, that's enough intuition for our purposes here. Now, it's not particularly useful to try to do direct minimization on the energy landscape because the dimensionality is still generally too high. Okay, and so if we simply try to minimize, that doesn't tend to work very well. So therefore, sampling methods that are designed to explore the conformational space, well, we want them to be efficient so that we can do a lot of these explorations efficiently in computational speed. And we also want to have good coverage of different low energy regions. And this is because we don't, it's very hard to narrow down specifically to one particular prediction. And so we want to, so it's useful to be able to come up with an ensemble of conformations that are at least plausible for what the real conformation might be from a low energy viewpoint. So that's what we call having good coverage of the conformational space. And so our approach towards this is from some of my earlier research was a sequential Monte Carlo method specifically designed for the protein, exploring protein conformations. And some of the key features is that we build the segments of the amino acid region one at a time, such that it is targeted, so the sampling is targeted towards low energy space. And it also takes advantage of this structure that I showed you on the board in terms of the relationship between the side chain and the backbone. And so a key concept in sequential Monte Carlo is what we call a particle population. So this is what allows us, via this method, to maintain a set of substantially distinct conformations, all of which are low energy and realistic from an energy viewpoint. Okay, so I will just give you some intuition. I won't go into all the details for the statistical aspects, but I do want to point out a few things. So there's one key distinction I want to point out between sequential Monte Carlo sampling methods and, say, Gibbs sampler. Okay, so in Gibbs sampling, we worked on conditional distributions. Okay, so for each variable, we sampled it conditional on everything else. So that's the idea of the Gibbs sampler. Now, in sequential Monte Carlo, we do it a different way. Okay, so instead of using a sequence of conditionals, we use a sequence of incremental distributions. Okay, so what we do is we first sample amino acid number 1. Okay, so we sample the angles for the first amino acid. And then for subsequent amino acids, we sample those angles conditional on the previously sampled ones. Okay, and so that's what's shown here in this first line. And so in order to do that, we also need to calculate how the energy changes as we're doing the sampling. And so here, this is the conditional incremental energy of x given previously sampled angles, which I've denoted x prime here. Okay, so as a schematic, you look at this part in the bottom. So if this is the segment that we're trying to explore conformations for, we're doing this one amino acid at a time using an alternating scheme. Okay, so we first here are shown the anchors. So this is the part of the protein that's not affected, and that we're going to leave fixed. Okay, so those are the left and right anchors for the segment. And so what sequential Monte Carlo does, it's going to add one amino acid to each end at a time to build possible conformations. Okay, so we'll add, say, one to the left, and then one to the right, one to the left, one to the right, and so on. Okay, and so that's how we can build backbone for this segment. Okay, so key idea in sequential Monte Carlo is to maintain a particle population. Okay, so so these are the collection of partially completed conformations. So at any time, we're going to keep a collection of n of them. Okay, so first we sample the first one. Okay, it's this first amino acid from according to a proposal distribution. Okay, so that's what I called eta 1. Okay, so we've seen proposal distributions in the context of, say, Metropolis Hastings. So here is sort of a variant of that. So we can sample from a distribution that is not exactly the same as the energy, and I'll explain why in a moment. So we sample from an approximated distribution. We can then adjust it by using doing a weight adjustment. Okay, so how we calculate the weight is the ratio of the actual energy, the Boltzmann distribution for the actual energy, so that's pi, divided by the distribution that we sampled from, the proposal, the importance distribution, eta 1. Okay, so this step continues as we propagate the particles forward, and in other words, as we build up protein segments. Okay, so at each time we sample the next set of angles according to proposal distribution, and then we update the Monte Carlo weights using these formulas. Okay, so how do we choose the proposal distribution? So it's much easier to evaluate the angle component than compared to the distance component. Okay, and the reason for that is, in order to evaluate the distance component, essentially we need to calculate all pairwise distances in space between all pairs of atoms. Okay, so it might seem like a good idea to simply use the Boltzmann corresponding to the angle energy as a proposal. But of course, if you ignore the distance, then we get a lot of atomic clashes. And also, such an approach doesn't make use of this geometric context, which is the relationship between the backbone and the side chain. Okay, and so what we do instead is we've adapted sequential Monte Carlo for protein segment, and there's three key contributions in terms of how key adaptations of sequential Monte Carlo to make it effective for exploring protein conformations. So they're listed here on this slide. I'll quickly run through them in just one or two slides each. Okay, so the first one is the proposal, and so how we set it up is essentially to discretize the possible angles and basically come and construct a list of angle pairs that satisfies a set of criteria. The first one is that these are realistic angle pairs seen in previous protein structures. Okay, so in other words, there is a non-zero density in the empirical distribution. Okay, so that's this region here that's highlighted. The second is that we want, as we put on as we sample the angles and therefore the atoms of the backbone, that it contributes a low energy, so the incremental energy is low, given all the other atoms that are in the spatial region where we are building the conformation. Okay, so that those are the atoms highlighted here. Thirdly, we want to check that there are plausible side chain conformations. So that's highlighted here in yellow that also have low energy conformations. And finally, we also want to check that we can also continue placing plausible conformations for the next amino acid. So for the next incremental sampling. Okay, and so that's a Monte Carlo strategy that we call look ahead. Okay, so that there's room to put the next amino acid in the conformational space. Okay, and so for the other angles, so that takes care of the two key angles, phi and psi. Omega is slightly more straightforward because there is a double bonded oxygen in the protein geometry. So omega typically has about 180 degrees as an angle, so there's not too much variability there. And finally, for the side chain angles at this point, we simply use a uniform distribution because we can embed a filtering step for that when we look at in the step that considers side chains specifically. Okay, so this sort of proposal is much better than simply using the angles. But if we draw too many, since we're constructing many of these draws for angles, how we should make good use of them in our energy calculations. And so that's the second that's the second adaptation that we've made for using sequential Monte Carlo for this method. And so from the proposal, we draw a lot of these potential continuations of the protein segment construction. And so say we generate a large number of them, let's say 100. Okay, so in other words, if we started with say 1,000, say we start with 1,000 potential conformations. So now we add in the next amino acid. If we have each of them can generate 100 more, so now we have 1,000 times 100 particles. Okay, so once we have these particles, the key idea here is we want to keep the most promising ones, in particular the low energy ones. Okay, so we downsample based on the energy function such that we achieved kind of two twin objectives, which is they have both low energy and they also have high diversity in the sense that we're not generating the same protein conformation many times. Okay, we want the conformations to be distinct. Okay, so as a thought experiment, well, what if we chose the subset uniformly at random? Well, we would miss out on a lot of the low energy ones because the particles would cover a large range of the energy spectrum. But if we go on the other hand, simply choose the ones that have the lowest energies, well, these are not completed conformations yet, so we might miss out on conformations that could lead to low energy conformations after more sampling steps have been done. So the statistical solution is to do stratified sampling. Okay, so we sample the lowest energy ones. So we take a whole bunch of lower energy ones, and then from the other stratum we sample uniformly. So that keeps a nice balance between ones that have incremental low energy and ones that could potentially be useful conformations for as we build the rest of the segment. Okay, and so the third innovation in our sampler is to handle sidechain particles. So I won't go into this in detail, but suppose here in green we have just added the next amino acid in our SMC exploration. So here is the dotted line is its sidechain. So we need to assess the impact of this possible sidechain conformations. Okay, so here are some possible sidechain conformations in the brown and the purple. And so if we need to assess how they perform energetically with the side chains of previous amino acids that we've built. And so basically we do a filtering step that assesses the interactions between the side chains that we're adding on our current amino acid with the side chains of the existing ones. Okay, and so that also helps stabilize the energy and reduces atomic clashes as we build our segment. So that summarizes the method. Okay, so so to recap, so here is a list of, here in this diagram, we have, say, here are potential conformations that we are building for, say, the mutated segment of the protein. Okay, so in the first step, we look at possible ways to add an amino acid onto the partially constructed conformation, and we examine the energy impacts of these additions. Okay, we identify what are the plausible conformations according to the energy criteria. Some of them might have dead ends. For example, in the third one, this might be an implausible conformation, and we weed it out. Okay, so from these plausible conformations, we end up with a much larger number of potential conformations. And so from here, this is where we do a filtering step and a subsample to get a set of conformations that are low energy and structurally diverse. And so that's how we can apply sequential Monte Carlo as a scheme for efficiently exploring the conformational space when we're particularly interested in the problem of looking at what are potential changes in structure when we target a specific region of a protein and look at amino acids to that particular range of amino acids. And so this is an overview of how we can apply ideas from statistical sampling methodology to help tackle one aspect, at least, of the data science problems that face us in terms of understanding uncertainty associated with protein structure. And so this is something that I'm continuing to work on in the coming days, and so I wanted to present this to you as give an idea of how we can take powerful sampling methods in statistics and apply them to current problems. Okay, so that summarizes and concludes the technical part of this lecture. And so just to wrap today up, I thought I would end on perhaps a slightly more upbeat and joyous note. So I'm going to shift my camera here in over here, and so I want to share with you just to end off a piece that's very famous, especially in America. So Gershwin was an American composer, and he wrote some pieces which are now classics in the musical repertoire, and so one particularly famous piece is the Rhapsody in Blue. Okay, so that was written in the early 20s, and so it was originally written for orchestra with piano, and so what I wanted to do here today, just to end off, is to share with you a performance of the solo piano version of it, and I really enjoy this piece because it's kind of it's a hybrid. It has elements of classical music and also jazz elements. And I also like it because it's pretty upbeat and kind of paints a picture of optimism. And so I hope you enjoy. Just give me a moment here to finish setting up my camera, and then I will head over to the piano to play this piece. Okay, so that brings us to the end of today's video, and so thanks for watching. If you enjoyed today's video, make sure you like, subscribe, and do stay healthy during this time. And I will now sign off. So all the best and cheers.